Good afternoon, everybody. This is Ellen Huckabee with the Alabama Cooperative Extension System, and welcome to our monthly webinar, All Bugs Good and Bad webinar. This month's topic is termites of the United States, biology, behavior, and management, presented by Dr. Robert Puckett of Texas A&M University. Dr. Puckett?
Thank you, Ellen. Good afternoon, everyone. As Ellen mentioned, I'm Robert Puckett, and I'm an assistant professor and extension entomologist at Texas A&M, and I'm with the Rollins Urban and Structural Entomology Facility. Previously, the Center for Urban and Structural Entomology at Texas A&M, where I did a postdoc and research scientist position with Dr. Roger Gold. And I actually did my dissertation research here at Texas A&M, and at the time I worked specifically with red imported fire ants. I saw some names from red imported fire ant land on the list of participants, so hello to you all. And I continue to work on those ants, but as part of my responsibilities when I became part of the Center for Urban and Structural Entomology at A&M, I was asked to work on termites, and it was kind of new to me, and so it's sort of a mountain of information that I had to climb right at first at the beginning of my position here, and I've really come to enjoy working with that group. But I continue to work with invasive ants as well, red imported fire ants, and most recently the tawny crazy ant, otherwise known as the raspberry crazy ant. But today we're here to talk about termites, and I should confess I've done all of my research during my graduate work and postgraduate work, and now in my new position with the university in Texas on insects that occur elsewhere, but all of my research has been done in Texas. And so there are some groups of termites that are really important in other parts of the South and the Southern United States that I'll briefly touch on here, but I think you'll see my Texas bias when we talk about termites that are really common in Texas, but many of those occur throughout the southeastern United States. And I should mention just briefly the format of the background of my slides has changed just a bit since I sent the talk in, so you might have to bear with me as I take a quick glance at the slides as we work through them. So we're here to talk about general biology, behavior, identification, maybe most importantly, of common termites in the South, as well as management. And the approach that we've taken over the years in building this talk is adapted from sort of one of our main presentations that we provide to pest management professionals in a couple of our training courses that I'll mention throughout the talk. So this presentation is adapted from that, and what I've done is try to just build in those aspects of termites, their biology, identification, and management, and embed components of each throughout the presentation. So I hope you'll enjoy how this flows. This is, in fact, my first webinar. This is my first time to do this, so it's a kind of a cool experience. So, termites. Turns out there's about 2,200 species of termites, and we know that they're most closely related to cockroaches, another insect that has a bad reputation for being a pest insect, particularly in urban and structural situations. And so those 2,200 species are separated, depending on whose taxonomy you find yourself aligning with, into 7 families. And in reality, the number of termites, or the number of species within the group of termites, is kind of small relative to some of our other insect groups. So if you think about like beetles, for instance, that have somewhere around 400,000 species, the termites are a small group, coming in at about 2,200 species. However, they're one of the most important insects that we deal with, both ecologically and economically. And I think if you sit through the remainder of this talk, I can convince you that this is in fact the case, if you're not already familiar with this. So termites themselves are small to medium-sized, soft-bodied insects. In particular, the workers and the soldiers, and the workers and soldiers are typically pale in coloration, And termite colonies are separated into a variety of different functional groups or forms or castes within the colony. And I'll just briefly mention the castes here. We're going to go through each of them as we work through the presentation and describe their role in the colony and their biology and behavior. So there's the king and queen, alates or swarmers, soldiers, and then surrogates or workers. So let's talk briefly about workers from an identification standpoint. So it turns out that when it comes to homeowners and folks that encounter infestations of termites, workers are the most common caste to be collected, and there's a good reason for that, and that is because there's more of those guys in colonies than any other caste. Typically far more workers than any other caste in the colony. The problem in terms of identification of one species from another using workers is that they're very, very difficult to distinguish between species using workers. There are no identification keys that are available, so this makes the situation very, very difficult because, like I say, these are typically the most commonly collected termites from folks that encounter them, like in their homes or in businesses, or find them in infesting structures. The next caste to discuss are the soldiers, and these are collected nearly as often as workers. It turns out that there's far fewer of these than workers in colonies. But they respond to challenges. So if you were to break into a wall void or a board that was infested with termites, particularly if you're dealing with some of our invasive termite species, the soldiers would rush to the site that was being challenged, and so they get collected too. And the good news is that they're better for identification than worker termites. So let me pull this arrow. Try to pull this arrow in so I can show you this. This is a soldier, this too. This happens to be a worker. And you can see that the head capsule of the soldiers is a dark, deep amber brown. They're hardened and sclerotized relative to the workers in the same colony. Also, they have these big conspicuous mandibles. Well, many species, certainly our subterranean termites, have conspicuous mandibles, and those are very important diagnostically speaking for identification of termites. And I'll work through this once we get to the sort of the identification section of the talk. So these happen to be Formosan subterranean termites, one of our invasive termite species here in the southern U.S., and we'll talk all about these just shortly. Yeah, so these can usually be identified, or termite colonies can usually be identified to the family level using soldiers. And as a result of their activity when a colony is challenged, and the fact that they occur throughout the year in the colony, they're the form that's very commonly used for identification. Well, so the best caste for identification are the alates, the swarmers. And this has to do with the fact that the pattern of wing venation on the wings of these guys is very diagnostic and can be very species specific. And we have very good identification keys that use the shape, size, and wing venation of the wings to help us identify them. The problem with these guys is that they're not available for collection throughout the year. So we're in sort of the swarming time for subterranean termites. Some of our subterranean termite species in the southern U.S. Some swarm a little bit later in the year. But once they've swarmed, by and large, it's very difficult to find alates. So if you were to find an infestation of termites of this particular species that happens to be swarming now in our part of the world, if you were to find that infestation just a few months from now, it's very unlikely that you would find swarmers, and so they wouldn't be there and available to use for identification. But if they are, if you can find these, they're the most accurate in terms of identification. So why do we care? Why do we concern ourselves with the proper identification of termites? Well, it turns out that management strategies for termites are not the same for every species. So we're going to talk specifically about subterranean termites and drywood termites over the course of the next 45 minutes or so. Those management of those termite groups are approached differently. So it's very, very important that we determine exactly what species that is found in an infestation. Family level identification is essential. In some cases, genus and species level identification is necessary to select the proper treatment and the proper management strategies for dealing with subterranean termites. Selection of the wrong management strategy can lead to a treatment failure. And if you're a customer, that's bad news for you. If you're a PMP, a pest management professional, that's bad news for you too, because your customer can find very easily and readily available ways of saying very bad things about your company in very short order on the internet, and that information can be shared very quickly. So it's good for everybody involved in a termite management situation to be acutely aware of exactly what species is being dealt with. So let's talk about some of the common subterranean termites in the southern U.S. and the species that I'm going to discuss are all in the family Rhinotermitidae. So these are some of our native subterranean termites, all in the same genus, Reticulotermes, and there's Reticulotermes flavipes. This is probably the most abundant species in the part of the southern U.S. where we live, in Texas. There's Virginicus, but as a matter of fact, I identified a series of specimens that a pest management professional sent in. I identified those just today as Virginicus, and they're down in southeast Texas. And then Hageni, Reticulotermes hageni. Well, we also have some invasive species in the same family, but these guys are in a different genus. They're the Coptotermes termites. Coptotermes formosanus. This is the Formosan subterranean termite, and then Coptotermes gastroi, the Asian subterranean termite. And you may have heard some very recent information, some very recent press involving the discovery of hybridization of these two species in southern Florida. So it's a really cool story, and you can go out and Google it and find it on the web right now. It looks like these two species, where they occur together in sympatry, have been observed to hybridize. And it's unclear whether or not that's going to create a much more difficult situation in terms of control, if their behavior is going to allow them to be much more of a problem in the areas that they occur, or if they'll actually kind of be wimpy. We'll see. Okay, so we receive specimens, like many of you guys do. Many of the folks that work for Extension receive specimens all the time from homeowners, property owners, pest management professionals, etc., that are concerned with an insect that they find in their structure or around their home or around their apartment, etc. One of the common groups that termites become confused with are ants, believe it or not. So we get lots of folks that send in specimens that believe they found termite swarmers, when in reality they've sent in specimens of ant swarmers. And in fact, those two groups aren't closely related at all. Termites belong to an order called Isoptera. Ants belong to the order Hymenoptera, with all the bees and wasps. So let me just kind of work through this slide. Looks like the format has changed a little bit here, but that will be easily manageable. So I just want to point out some of the conspicuous differences between termite swarmers and ant swarmers. So ant alates and termite alates. And maybe the most conspicuous difference is this constriction that occurs in our ant swarmer between the thorax and the abdomen. You'll note that our termite swarmer is much more broadly joined between the thorax and the abdomen. So the waist does not occur in our termite swarmers. And the other big conspicuous difference are the size and shape of the wings. So like in our termite here, you'll note that the forewing is roughly the same size and shape as the hind wing, whereas in our ant swarmer, the forewing is much longer than the hind wing. And another good diagnostic character is the shape of the antennae. So with ants, their antennae are elbowed. So they have this long segment coming off the head. We call that the scape, and then a series of segments extending from the scape. And the number of segments is actually very important when you're identifying different species of ants. So keep this in mind if you don't already know this. If you use an identification key to identify ants, be it workers or swarmers, very quickly you'll come to a couplet that describes the antennae and the number of antennal segments. Always remember, count the scape. The scape counts as segment number 1. It's actually a segment of the antennae. And this is a common mistake that people make when they first start working with identification keys for ants. They forget to count this segment, and it send you the wrong direction in an identification key. So anyway, back to our termites. So ants have elbowed antennae. Termites do not. They have a series of segments. They take on, not in this depiction, but in real life, they take on the appearance of a string of beads. So the antennal segments are very rounded and kind of look like a string of beads. So many of you, and usually at this point in the talk, I'll ask for a show of hands how many folks have used identification keys for identifying insects, and usually the response is just a couple of hands that go up in a room full of 100 or so people. Most folks don't use identification keys, or maybe they have and don't realize it, but I think in most cases people are unfamiliar with them. So I want to work through them just briefly because they're actually a very, very important tool for identifying insects or any other group of insects or group of animals, or organisms, I should say. There's plant identification keys as well, of course. So typically keys occur in two types. So they're either picture keys or written keys, but the way that these work is that in this case, your insect, the specimen that you're working with, you work it through a series of couplets. And a couplet is binary, as you'll see, and each couplet contains one set of characters or a single character for identification. And if you get to the end of the characters that are being described and you find a number at the end, as you'll see on the next slide, it means you go to that couplet and you work through that next pair of binary characters or keys. Or if you find a name at the end of that set of characters, then you've found your diagnosis. You've identified your specimen. So I'll give you an example of this and how this works. For those of you that have not used identification keys, this is a written description key, and this is a key to families of termites in the United States. So we'll imagine that we've got a specimen in front of us, and we look at our specimen, and then we go and read this set of characters, and it says the anterior of each wing has 2 heavy veins. Those heavy veins on the wings are very important, and we're going to get to that just shortly. The fontanelle is usually present. Clypeus is bilobed or divided by a line. So now we look at our specimen, and let's imagine a scenario where our characters don't really match up with this description. So we simply go to the next step here in this couplet number 1. Anterior of each wing with 3 heavy veins, and it turns out our specimen does have 3 heavy veins on its wings. So at that point, we determine which couplet we're going to next, and it's number 3, and you'll see number 3 ends. There's no way out, so you're going to arrive at a family name here. And usually at that point, the next step is to go and find a key to the genera or the species that occur in that family specifically, if you need a genus or species level identification. Okay, so the other type of keys that are commonly used are picture keys. Works really the same way. We work through the key, we compare our specimen to the pictures here. So let's imagine ours does not match this picture. We would go to this picture, and hopefully your specimen matches this guy, and then we'll go to the next set of pictures. And on until we finally arrive at the end of the key, at an identification. So these are very, very important tools, and I use them every day. And if you're a PMP, you should try using these too. I'm sure our extension agents and specialists all have different keys for different organisms, different groups of insects. They're very, very important for what we do. But they can be problematic. So first of all, you got to know how to use keys, and you just learned how to use them. The next step, or the next problem that you can have on is that you got to be able to recognize the characters that are being evaluated. So I mentioned a couple of characters in the last slide, the fontanelle and clypeus. Well, if you don't happen to know what those characters are, you spend a lot of time in the glossary of terms, and that goes for all of us. Like the first time that I use a new key for a group that I'm not very familiar with, I find myself doing a lot of that, checking the glossary of terms for the characters that are being described. But with some experience, you gain a familiarity of those characters. Yeah, and so the other thing is that the characters on a specimen may look very different from a picture or drawing. And I always ask this question, folks in the audience, like if you were told that that you needed to develop a photographic key for some insect group, would you use the best or the worst images that you could find? And the reality is you would likely find the best possible images that you could of that insect. Well, in reality, most of the specimens that we have to compare against those photographs are not in as good a shape. So this creates a challenge as well. But this can be overcome with experience, and sometimes you seek out somebody with expertise in a group that you may not have. And I always make it clear to everybody, I'm sure that the county agents and extension specialists in your state, wherever you guys happen to be, are very willing to help. Like I mentioned before, I identified termite specimens this morning, and just about every day I receive specimens in the mail, and we're happy to do that work. And so we try to make folks aware that we're a resource. And whether or not you happen to find yourself in the state of Texas, we're a resource for everybody. If you need help identifying any group of insects, we'll be happy to help, especially those that are urban and structural importance, like our termites. And so this slide contains all of my contact information. I'm sure this is being recorded, so you can go back and take a look if you need to get this later. But I'm going to move on. Oh, I should mention. So this is our brand new laboratory. We just moved into this about a month ago, and it's just as beautiful as it appears on the screen. It is just a fantastic facility. I wish I had time to show you lots of images of it. We're very, very excited. Okay, so if you do need help with identification, you want to send specimens in to specialists, do them a favor and drop them into a leak-proof container with some alcohol. These happen to be insect vials. You don't have to use something so fancy. Any leak-proof container will work. And I showed you our address before. You can send those to us or somebody in your state. Drop them in a vial or a leak-proof container with 70 to 80% ethyl alcohol or isopropyl alcohol. If you can collect more than one specimen, that always helps. Labels are absolutely critical. We're the land-grant institution in the state, so we consider this part of our service to the folks in our state. And the only thing that we charge in return is good information for where those specimens were collected, because we keep those, and those help to populate our insect collection on our main campus. Please do not. There's a number of things that we ask you not to do if you do send in specimens that you probably find yourself, or for those of you that receive specimens from folks, that you find yourselves asking them not to do. So if you happen to be a PMP or a county agent or an extension specialist, you've probably received specimens like this. These happen to be fire ants that somebody thought it would be a good idea to press them between Scotch tape. And invariably, those end up underneath 800 pounds of mail as they get shipped to you in an envelope, a paper envelope. And then you're challenged with either trying to look at them through a microscope through that tape, or peeling the tape open. And of course, this guy's legs end up over here, and this guy's head ends up here, and it's a very messy situation. And can be even messier. So these happen to be termite soldiers that were sent in and got squashed underneath some mail. So this becomes sometimes, in some cases, a nearly impossible situation in terms of identification of specimens that arrive in this manner. So one thing that we have begun to suggest to folks, if you are in a position that you're somebody that receives specimens, you likely receive lots of photographs of insects. And you're likely aware that in most cases it's very, very difficult to make an identification from photographs. And that has to do with the fact that a lot of times the cameras that we use to take photographs of insects simply aren't equipped to take very good images of very small subjects from a very short distance. But it turns out that most of us in our pocket or around our desk or wherever you happen to find yourself right now have a smartphone in your pocket. And that phone likely has a camera embedded in it that's better than some of the best digital cameras from just a few years back. And if you use those devices and couple them with something very, very inexpensive like this jeweler's loupe, you can create a very inexpensive macro photography setup. And this is something that we've really been promoting among our PMPs, because we get lots of photographs from the field, and unfortunately, most of them are not usable from an identification standpoint, particularly with termites. So Alex Wild, his blog, many of you know or know of Alex. He's become a very famous insect photographer, an academic that became very, very good at taking very, very just exceptional insect photographs. And we read on his blog a couple of years ago, he told the story of using magnification like this coupled with a phone device like this and the camera embedded in the phone to take really good photographs of small insects. And so we've really been encouraging folks to try this, and I encourage all of you to just give it a whirl. It's actually pretty fun, and you can get some really, really cool images. So this is an image of an insect that's pinned just with my cell phone camera. This is at full magnification with the camera in the phone using the loop method that I just showed you. The loop is just held over the lens of the photograph. Oh yeah, Tim Davis reminds me, Alex Wild is now at the University of Texas, where he moved here recently from University of Illinois, and he's working now at the University of Texas in our state. Let me point out, so you saw this insect in the image before. This is at full magnification. And the camera, I was just holding the camera with my hand at the time. I steadied myself against the table, and you can literally count hairs on this insect in this image. So I really encourage you to try that if you are somebody that routinely receives photographs from folks. Tell them about this method. If you're somebody that routinely sends insect photographs to folks, give this a whirl. I think you'll be pleased with the result. And some folks have actually become fairly artistic in the images that they've taken with this method. Okay. So let's talk about the biology of subterranean termites. And this is just in a general sense, the biology of in particular, our subterranean termites. We'll talk about drywood termites just shortly. Okay, so you are all likely aware that termite management is a major focus of the pest control industry, both here in the United States and abroad globally. But it's good to keep in mind that in nature, termites are beneficial insects. So they're part of a decomposer community that's responsible for breaking down plant material as it dies and decays, and thereafter cellulose, a component of plant cells. That's their chief source of nutrients. So in nature, the decomposer community is a vital part of properly functioning ecosystems, and so these guys are responsible for part of that work. They're some of the first organisms that begin to break down, in particular, woody plants when they die, when limbs break off, or when whole trees die and fall to a forest floor. But the problem is they don't really know the difference between a log on a forest floor or a 2x4 that's supporting part of a wall in our houses or the structures that we value. And they're present throughout most of the United States. So subterranean termites do a great deal of economic damage as they overlap with human populations. And so the question is, why are they such pests? And why have they become such a key component in the pest management industry? Well, they're able to live for many, many years as a colony, as you'll see shortly. And their colonies tend to be what we refer to as self-perpetuating, meaning that there are cast members within a colony that if they happen to become cut off from the main colony, there are members within them that can become supplemental in terms of their reproductive activity. They can lay eggs, and if that portion of the colony ever makes contact with the main colony again, they can just mold right back into the main colony. They have a virtually unlimited supply of food, both in cellulose material that occurs naturally in the environment and that which we provide for them. And they live underground, and this is really an important aspect of their biology. Living underground protects them from environmental extremes, cold and heat and hot situations. Not so much flooding situations, but heat and cold extremes. And the other interesting part of their biology, I'll just briefly go through this slide. It looks like the format changed a little bit when it was converted. So if you think about a general insect life cycle, imagine, for example, bed bugs. So a male and a female bed bug, they pair up, they mate, the female lays eggs, and those eggs hatch at some point, and the nymphs develop through to adulthood, and the cycle begins anew, and it's all very circular. Well, with termites, it's a bit different. So or there's aspects of their life cycle that are different from that which I just described. So you've got king and queen that pair and mate, and then the queen lays eggs, and those hatch, and the very young larval instars begin to develop through. And depending on the needs of the colony and the feedback that the colony is getting, they can become pre-soldiers, at which point they're fated to become soldiers, the defense force of the colony, or they can become workers or surrogates, some of which can become supplementally reproductive if the need arises. They can become nymphs. And typically nymphs are on their way to developing into alates or swarmers, and that's sort of the natural way that new colonies are made. The swarmers leave the colony at some point and mate with individuals from other colonies and begin a new colony of their own. But some of the nymphs can also become supplemental reproductives in the colony if that becomes necessary. If a queen happens to die, or if, as I said before, they get cut off from the remainder of the colony as they forage and make their way through a subterranean environment. Okay, so let's talk specifically about the castes in colonies and their behavior and biology. And so we mentioned before there's a variety of different castes, larvae, surrogates, soldiers, nymphs, and reproductives. The number of each type within a colony is regulated by feedback of pheromones to the queen. And so the first group to talk about here are the larvae themselves. So these are very young termites, the first and second instar. Or stage of development. These guys, if you encounter these, you'll see they don't have wings or developing wings. They're very sort of simplistic in their body plan relative to some of the other castes. One thing to mention is they can't feed themselves, and for that reason, they're sort of an energetic drain on the colony. But the trade-off is that these guys happen to be sort of the next labor force, the labor-ready force in the colony. So surrogates and workers. So these guys, this is sort of the business end of the colony. These are the insects that are responsible for carrying out the work of the colony. And they're the ones that are responsible for doing damage to wood. And so you can see these guys happen to be on some tongue depressors in one of our laboratory colonies. But the reality is these are the insects that are chewing for sort of the stomach of the colony. They're the ones foraging on wood sources. Okay, soldiers. So these are sterile members of the colony, both males and females. And as I mentioned before, they have a large, well, in this case, elongated and highly sclerotized or hardened head relative to like the workers that I just showed you. Many groups have very large mandibles. Our subterranean termites have very conspicuous mandibles. These are adapted for colony defense. Now, usually at this point in the talk, I ask if folks are aware of the major natural enemies of termites. And of course, you guys can't answer directly, or else I can't see your answers, but those are ants. So their main natural enemies are ants. And so these mandibles are adapted for defending colonies against challenges from ants. And like our larvae, I mentioned before, these guys are incapable of feeding themselves. So the workers in the colony take on that duty and feed these guys. And of course, the trade-off is that they're the defense force of the colony. So I'll just briefly mention nymphs. These are sexually immature termites that are on their way to becoming sexually mature, typically in the process of becoming alates. And as I said before, they can become supplementally reproductive within the colony if the need arises. And when they're observed in a colony, some are younger than others, and the younger ones typically have very short wing buds that are emerging. They get longer as they grow, so they can be even wingless, and in some cases with short developing wings. Okay, and then there's the primary reproductives, referred to as the king and queen of the colony. And these guys, of course, are the result of swarmers from the previous colony. So these are the insects that are responsible for starting new colonies through mating flights. And often these are the first insects, in terms of an infestation, that are noticed by homeowners. So the reason I say that, if you've ever been out in a forest or outside and seen a big swarm of subterranean termites, those that swarm during the day, it's a very conspicuous event. So hundreds of swarmers leaving a log. They're kind of poor flyers. They kind of float around through the air, and it can be a fairly dramatic event. Now, if you imagine that happening inside somebody's bathroom or a kitchen, This is an event that homeowners tend to not overlook, and they don't forget. Yeah, so swarmers are typically, in terms of our subterranean termites, black or dark brown in color. I mentioned the sclerotized heads of soldiers, where, well, the whole bodies of the alates or swarmers are sclerotized and hardened. Okay, so swarming itself. So this is the process by which new colonies are typically started, and depending on which species you're dealing with, these occur at specific times of the year. Typically, in our part of the United States, starting in January, late January, through April. In most species during daylight hours, but the Formosan subterranean termites that I'll talk about shortly, they swarm at night. That's a very conspicuous part of their behavior. So the number of alates that leave a colony, the number of alates that are present in a colony is typically proportionate to the colony's age and maturity. And we'll talk about how colonies age and grow just shortly. It turns out that very few swarmers live to reach maturity and live to start a new colony. So they're one of our typical K-selected species of organisms where there's lots of eggs and lots of larvae, but very few of those make it through to adulthood. Fortunately for us in our homes, I might add. So in terms of nesting sites, so now we're starting to move towards thinking about control and mitigation of termite infestation. So this is a very, very important aspect of subterranean termite biology. These guys basically have two necessary components of a proper nesting site, and those are soil and moisture. These guys require a constant source of moisture to keep from drying out. So if you think about these guys, they're soft-bodied insects, at least the workers and the soldiers. They have a high surface-to-volume ratio, and the reality is they'll just dry out very, very quickly. So if I were to place some in a petri dish that was open inside the lab and leave them out overnight, they would likely be dead tomorrow morning as a result of desiccation. So they have to stay moist all the time. And in fact, many of our subterranean termite species, our native subterranean termites, have to use mud tubes. We call them mud tubes. They build earthen tubes when they leave the soil and forage onto structures. And so this is another aspect of their biology that makes them semi-easy to spot when they're infesting a structure. So as they move from the soil onto the wood inside or on the outside of a structure, they typically build a mud tube that they forage up and down inside, and this allows them to keep from drying out. Well, those mud tubes are a very telltale sign of termite infestations. But one thing to point out is that when we talk about them requiring a constant source of moisture, it doesn't necessarily mean that there needs to be a flood in a house, right? There doesn't have to be that much moisture. The moisture that is required to support a population and an infestation of subterranean termites can result from minor moisture sources, so like a leaky roof or leaky pipes, shower stalls, even condensation that collects on pipes and runs down and collects in a particular area of the structure, even humidity. So it doesn't take a lot of moisture, but it requires a constant source of moisture. Okay, so in terms of colony development and in terms of the lifespan of termites, folks are usually surprised to know that the king and queen, the reproductive unit in the colony, can live very long periods of time, upwards of 20 years in some cases. They take a few years for colonies to reach maturity, whereas soldiers and workers are much more short-lived relative to kings and queens. They live just a short period of time, 2 to 3 years. During the first few months of a colony, the queen's laying just a small number of eggs per day. And the reason for that is that those insects, as they develop, have to be cared for by the king and queen. But at some point, they complete their second molt as immatures and are able to feed themselves and siblings at some point. So during the first few years of a new colony, all offspring will either be workers or soldiers. But as I said before, as the colony grows, care of new offspring is delegated to older siblings. And so the more older siblings that you have, the more offspring that the colony can support. So a mature colony, these are ones that all casts can be found over the course of a year. So nymphs, swarmers, soldiers, workers, kings, queens, and colonies can be very large, in particular when we talk about our Formosan subterranean termites, over 1 million termites in some cases. Well, so the time that it takes for a colony to reach maturity depends on a number of things, food availability, all of the resources that they acquire, and those resources availability. So food availability, shelter, environmental conditions, etc. So I mentioned before, cellulose is a food source for all termites. That's what they're after. That's what they feed on. And those nesting in the ground can use their mud tubes that I mentioned before to reach sources of cellulose that happen to be up in a structure, for instance. One thing that's very interesting about their biology is they require mutualistic microorganisms, protozoa and bacteria that live in their hindguts to help them break down the cellulose. They don't have the enzymes to actually break down cellulose that they feed on. They require microorganisms in their gut to do this work that do have the enzymes. And so it's important to point out, as they molt through in development, they lose those microorganisms each time they molt. And to get them back, they have to engage in this behavior called proctodial feeding. So this is feeding on anal secretions from other termites that have a full complement of gut microorganisms. So, you know, it's kind of a gross and tough way to make a living, but they're actually very, very good at it. So I mentioned before, the workers are responsible for gathering food for the colony. And we say that they're not selective in terms of the type of food that they eat. Well, so they'll feed on all sort of forms of wood that they find in their environment, but there do seem to be some preferences, and I'll talk about this as we discuss Formosan subterranean termites in just a couple of slides. We'll begin that discussion now. So many of you, if you're in the southern United States, you live in a state where one of our really important invasive termite species occurs. And these are the Formosan subterranean termites, Termites formosanus. And so these are likely the most destructive termites to infest structures and urban systems in the United States and elsewhere. So they're native to Southeast Asia, China, Japan, and Taiwan, which of course was formerly known as Formosa. That's where they get the common name, the Formosan subterranean termites. And we think that they were introduced to the southern U.S. From the Pacific theater after World War II. And the idea is that they came back infesting wooden items that came back from that theater. They were first identified in the United States in 1956. And they belong to the same family of subterranean termites as our native subs, the Rhinotermitidae. Remember, I mentioned this in a slide earlier in the talk. Okay, so I've lost my green arrow, and I don't see that here. Ellen, can you hear me? Well, I'll keep moving here, and maybe we can recover that, and I'll try and just direct your attention to what I'm discussing and trying to point out verbally until we recover that arrow.
It was there a second ago.
I'm going to break from the presentation real quick and talk to Ellen. I don't think you guys can hear her. Ellen, I'm sorry?
It's right there on the left-hand side of the screen. Can you see it?
I cannot see it.
Oh, it keeps coming up and then going away.
Huh. How about now?
It's on my screen.
Well, I can't see it on mine, so I'll just keep moving through. So now at present, we have these termites are known infestations of this invasive species in 31 Texas counties, including, it might be difficult for you to see, but there's a county in the center of all that red, or actually it's in the center of a sea of white counties, all by itself. And that's actually Brazos County. That's where I sit just now, home of Texas A&M University. That's our home county. And those termites, the Formosan termites, were discovered here in 2010, infesting a neighborhood in College Station, Texas. And it's kind of an interesting story. There was actually a person who worked on termites, a man named Grady Glenn, who did his PhD work on Formosan termites, and he actually happened to be outside one night in the spring of the year, that year in 2010, and he saw these termites swarming in the lights around a department store. And so there was a press release on the following Monday, and so anyway, we were contacted. We went in and kind of went on the hunt for the infestation of formosans, and for a couple of nights we kind of hung around some lights, because remember I said before they're night swarmers. We hung around some lights in that same area. We didn't see any, and we actually gained access to a swimming pool in an apartment complex near there and found some swarmers in the pool filters. And so we talked to Grady, and he told us what night exactly it was that he saw them. And so we looked at the weather report from that night and saw which way the wind was blowing and pulled out some maps and just kind of backtracked and drove into this subdivision. And there were railroad ties that were supporting around the soil in a roundabout area, and all of those railroad ties were railroad timbers were completely infested with formosan subterranean termites. And so railroad timbers are actually a big culprit in terms of how these insects have been able to expand their range in the southern U.S. I didn't mention it earlier, but mark recapture studies of swarmers of these termites indicate that they're actually pretty poor flyers, or that they don't care to fly very far during their nuptial flights, just a mile or so, 2 miles at most. And so it's really hard to imagine this level of range expansion over the past couple of decades as a result of mating flights that are 2 miles at a time. The reality is we move these insects around like many of our invasive insect species. And these railroad timbers are a big culprit. We know that to be the case, as well as any other organic material that these guys can come in contact with and call home. You see the mulch on the right-hand side of the screen. That they will begin to infest bags of mulch that are left on pallets for an extended period of time in formosan termite-infested areas. And then if a homeowner takes those home, well, of course, they can just they can carry those termites unknowingly into their structure. And that's the case with the image that you see on the right. You'll notice just behind the bag, there's a mud tube emerging, and it moves towards the wall. And actually, that looks to be how this house became infested with formosan subterranean termites. Okay, so a big important part of their biology, and one of the things that makes them more difficult to control than many of our native species of termites, is that they don't require constant contact with the soil. So they're subterranean termites by nature, but they can move out of the soil and into a structure or into a tree. One thing I haven't mentioned yet is that these termites will actually infest living and healthy trees. And they'll do so in the heartwood of the tree. They'll begin to feed on the interior of a tree, and you're likely aware that nutrients flow up and down the outside of the trunk of a tree underneath the bark. Well, the termites forage from the inside out and begin to core out the center of the tree. But because nutrients are flowing properly, the tree behaves like a healthy tree, physiologically speaking. And so during the spring, it flushes leaves. It flushes leaves in the same amount of biomass that it would if it wasn't infested. But all the while, the termites are creating a hollow space inside the tree. And so you can imagine at some point, like in the spring, if you get some straight line winds that are associated with storms, trees can fail. They can buckle under their own weight, and this can become a significant liability for homeowners. You don't want trees falling down on your property and damaging your property or somebody there, or certainly not your neighbors. Formosan termites have a much higher reproductive potential than our native subterranean termites, and that's another important aspect of their biology that makes them difficult to deal with. And they'll infest and attack a wider variety of materials. So in terms of their identification, the workers or pseudorgates, they're off color or to white in color, just like our native subterranean termites. They're slightly larger than our native subterranean termites, but you have a very, very difficult time telling them from our native subs if you weren't looking at them side by side. So they're essentially the workers are essentially useless for identification, but the soldiers are not. The soldiers are actually very easy to identify compared to, well, to identify as being different from our native subterranean termites. So they have teardrop or egg-shaped heads that tend to be shorter than the heads of our native subs. And you'll notice, so the Formosan termite is on the right in the picture that you see on your screen. You'll notice the typical teardrop-shaped head capsule as compared to our native subterranean termite, Reticulotermes flavipes, the one on the left-hand side of the image. You'll note the sort of the parallel lines on the right and left side of the head of that termite. It has a very blocky or squared-shaped head compared to our Formosan subterranean termite, and they're much more abundant in the colony than our native subs. So they come from a part of the world that there's lots of tropical species of ants that occur there, and so they're very aggressive in terms of their defense. In some cases, if you disturb a colony of native subterranean, for flavipes, for instance, one of our native subterranean termites, it may take a while for soldiers to attempt to defend the colony against that challenge, but not with Formosan termites. The soldiers are much more plentiful, and they're very, very aggressive. They'll show up at the site of challenge much more quickly than our native subs. So in terms of the reproductives, the swarmers, well, they're slightly larger than our native subs. They're honey brown in color. They're much less dark in coloration than our native subterranean termites. But beware, some of our drywood termites tend to be the same color as Formosan subterranean termites. One thing to mention that's very important is that the wings of Formosan termites are covered with very fine hair. And you can see these with magnification. I think so in this slide, I want to demonstrate the difference. So there's two images on your screen. There's one on the left-hand side and one on the right-hand side. Let's focus our attention on the left-hand side. And you'll see that the top image is of a Formosan subterranean wing. The bottom image on the left-hand set of wing images or illustrations is an eastern subterranean termite wing, one of our native subs. Note, first off, that there are two very conspicuous veins on the leading edge of the wing. And then compare that to the middle image on the left-hand side. That's of a drywood termite wing. Note that they have three main veins coming off from the body and extending along the leading edge of the wing. And then out at the end of the wing, you'll see that there are cross veins that are present that don't occur in our native subterranean termites. But you'll also notice the stippling on those drawings, the little dots. Those represent hairs. And so if you look at the top image, that Formosan termite wing is covered in hairs, whereas the distal portion of our drywood termite has hairs, and then the bottom sketch there, the eastern subterranean wing, has no hairs at all. Now, if you'll move your attention over to the images on the right-hand side and go from top to bottom, you'll see the same scenario. These are in the same order as the wings on the left-hand side. So we've got our Formosan subterranean termite at the top with two veins and lots of hairs. I think you can see those. At the bottom, our native subterranean termite, two veins but no hairs, and in the middle, our drywood. But that's the portion of the drywood termite wing that's very close to the body, so you can't really make out the hairs. They don't start till a little bit further out on the wing than that which is pictured here. But you can see the three veins that are present. And I should point out, if you collect alates, or if you're given a specimen of alates to identify, pluck a wing off of one of them. Put it on a piece of glass. If you happen to have microscope slides, awesome. If not, any clear piece of glass will work. Drop a little bit of alcohol or even water on the wing, and then press it with another piece of glass, another microscope slide or another clear piece of glass. And what that will do is force those veins apart. Because one of the mistakes people make when they're identifying these is the veins tend to fold up on top of each other, and it's very difficult to determine if you don't do what I'm saying and press them between two pieces of glass. It's very, very difficult to determine accurately how many veins you're actually seeing. Okay, so swarming and mating usually takes place in late spring. We call these guys Mother's Day swarmers. And going back to that story that I mentioned before about Grady finding Formosan termite swarming in College Station, what I did mention is that it was a Friday night that he saw those swarmers. That next Sunday morning was Mother's Day. So their swarming activity tends to sync up with Mother's Day in our part of the world. It's the time of the year that they receive the environmental cues to mate. So I mentioned before, swarming with formosans takes place at night. Courtship and pairing is similar to that of our native subterranean termites. One of the things that's different, and this is, I mentioned before that they will move the entire colony in some cases into a structure or up into the trunk of trees that they're infesting. Well, they use this behavior to do that. We refer to this material that you see in the image here as carton nesting or carton nest. And it's made of chewed wood and all bound together by saliva and fecal particles. And really, it reminds me, I mentioned before, I work on fire ants as well. It really looks like, if you ever see an old baked fire ant mound out in a pasture and you cut it open, you'll see tunnels and labyrinths that look very similar to this, and it's actually used in a very similar way. But they can build this up in wall voids, in attics, above ground, of course, in a structure. And the problem with this is that they can, if they have a source of moisture, and they have a source of food in the form of wood that's used to build a structure, and if they choose to move the colony up and build carton nesting inside a colony, they can move the entire colony there, and they can forage for long periods of time in a very inconspicuous manner. So if this happens to take place in an attic of a home, unless somebody's inspecting for them, often they can persist there for long periods of time and do lots of damage to a structure before they're discovered. Yeah, so they require a few years to reach maturity, and during the first few years, their colony grows similar to those of our native subterranean termites. But typical mature formosan colonies are very large, several 100,000 individuals to over 1 million, and as I mentioned before, they have great reproductive potential. So in terms of management of subterranean termites as a group, not necessarily formosan termites, I'll mention those specifically as we work through the next slides, or the next few slides. The best course of management for subterranean termites is to not have them infest the structure in the first place. So preventative treatment is obviously always the best management practice for any insect group, but in particular termites. So this requires pre-construction application of chemical termiticides, a horizontal barrier across the area where the foundation is to be laid on a structure, and then of course the concrete is poured over that chemical barrier that has been created. Now the other thing to mention in new structures that should be built, or even in old structures, the key is to make sure that siding and insulation are placed above the soil grade, or where the soil meets the foundation should be lower, or the soil level itself should be lower than where the siding meets the foundation. Because typically subterranean termites will tunnel up the side of a foundation, the concrete side of a foundation, then they'll meet the brick or wood construction or siding and move into the structure, either through weep holes or through crevices in either the brick or the siding or the wood. And if the soil happens to occur above that interface where the siding, brick, or wood meets the foundation, it's no longer necessary for subs to build those mud tubes, and as a result, you don't have the big conspicuous characteristic mud tubes that are often used to identify infestations of subterranean termites. Okay, but if found, if pre-treatment doesn't solve your problem, or if after a home is constructed even with a pre-treatment barrier, and termites make their way onto the structure, there have to be mitigative treatments for dealing with the infestation, and that requires building a chemical barrier around the area that's infested, either through the creation of spot treatment barriers with chemical termiticide, and so in this case, a trench is built 10 feet of treated area around the infestation or the site of infestation. That material is applied to the soil, termites forage through that, and eventually the idea is that kills the colony. But with formosan termites, it's probably a better idea, if formosan termite infestation is identified, it's probably a better idea to treat the entire perimeter of the structure. And that's a very aggressive treatment, but called for in cases of formosan subs. Fumigation is a tool that's used for dealing with termite infestations, but with subterranean termites, it has to be used in conjunction with chemical barriers. And the reason for that is if you imagine a home that's infested, say, with Formosan termites, and they happen to build a carton nest in an attic or a wall void, they're no longer connected to the soil outside, but you know that there are likely termites on the outside of the house foraging as well, be it of the same colony or a different colony. A fumigation will simply kill the termites that occur in the house at the time the fumigation is made. And when it's safe for the homeowners or inhabitants of that structure to move back in, it's also safe for new termite infestations to move in, and they'll likely happily do so. So once a fumigation is undertaken, it's almost a requisite to make a chemical application on the outside of the structure as well to prevent future reinfestation. And then there's baits for controlling termites, and these work very simply by placing bait stations in the ground, a source of food in the form of wood is added to those bait stations. Once termites are found foraging on the wood is replaced with a chemical termiticide that's applied to a substrate and then inserted into the bait station, and those termites feed on that material, return it to the colony, feed their nest mates, and eventually reduce the population of termites that are foraging on the structure. And then with Formosan termites, often it is the case that folks have infested trees on their property. Those can be dealt with. And in fact, it's a good idea to do so because those populations of termites that are infesting a tree, this infestation can become a source population of termites that will gladly move to a structure nearby. They forage great distances in their foraging activity, and so the way this is usually conducted is that a tree that's identified as infested, holes are drilled into the tree and chemical termiticide is applied directly into the cavity where termites are infesting the tree. Often foaming agents are used to carry the termiticide up and down the cavern so that it contacts all of the termites that occur in the tree. Okay, so I got a message just a couple of minutes ago that I've gone a bit long, so I think I'll stop now and then I'll be happy to take any questions that you have.
We do want to thank everyone for attending today, and thank you, Dr. Puckett, for a great presentation. We do have 4 little survey questions that are going to pop up on your screen, and we would love for all participants to take those really quickly. And then we also have a link to a Qualtrics survey. It's just 11 questions to quickly get some feedback and other programs you may be interested in attending in the future. So thank you for attending.
And thank you all for your attention.
Dr. Puckett, are you still there?
I am.
Hey, thanks